Hi everyone, this is Sydney Wilson. Today I'm going to be presenting a little bit about the movement of macromolecules within the cell. We're going to touch on three different macromolecule types, first being a lipid, then being a secreted protein, and then an intracellular protein. So we are going to get started with the lipid. Specifically, we're going to talk about estrogen, which is a steroid hormone. We're going to focus on the production of estrogen within the follicles of the ovaries. This process is going to begin with the uptake of LDL cholesterol from the bloodstream by the thecal cells of ovarian follicles. The LDL is then transported to the smooth endoplasmic reticulum by transport vesicles. From here, enzymes in the smooth endoplasmic reticulum use cholesterol to produce androgens. Androgens then diffuse to nearby granulosa cells where they meet with aromatase. Aromatase is an enzyme that is crucial to the production of estrogen, but it is not present in the thecal cell, so this is why the androgens have to diffuse into these nearby granulosa cells instead. Once it does reach the granulosa cells, aromatase is going to convert the androgens into estrogen in their smooth endoplasmic reticulum. After that, estrogen is going to be packaged into secretory vesicles and taken to the plasma membrane to be diffused back into the bloodstream as estrogen via exocytosis. From here, estrogen is important in carrying out functions like menstrual cycle regulation, libido stimulation, reproductive organ growth, and many other functions as well. Next, we're going to talk about a secreted protein. The one we're going to focus on is pancreatic amylase. The production of pancreatic amylase is going to begin as messenger RNA delivers transcribed instructions to the ribosomes in the rough ER of these acinar cells. From here, transfer RNA is going to assist in the assembly of individual amino acids required to build the pancreatic amylase molecule. The amylase enzyme is then packaged into a transport vesicle and is sent to the Golgi apparatus. The Golgi apparatus sorts and packages the enzymes into transport vesicles now. These vesicles are exported to the cell membrane where amylase will be released into the pancreatic duct. The pancreatic duct will drain into the duodenum of the small intestine where the amylase will assist in the breakdown of carbohydrates. This enzyme is super crucial to the whole digestion process that is going to take place within the small intestines. It is especially important for the breakdown of large carbohydrate molecules into smaller monomers that can be absorbed. Next, we are going to talk about an intracellular protein called actin. The production of actin is going to take place in the cytosol at or near the cell's membrane when a small aggregate of 3 actin globulin monomers is spontaneously formed to create what's called an actin nucleus. This is the first step of the process and is also called nucleation. From here, we're going to move on to the elongation phase, which begins as additional actin monomers polymerize both ends of this original trimer and is going to form a much longer chain. From here, and that's considered the elongation phase. From here, we are going to move into the next phase where the actin filament reaches a steady state. It is no longer attracting globular actin proteins for further polarization. Once the steady state is reached, the actin filament will be able to join the cytoskeletal complex of the cell membrane. It is going to be a super important protein in things like motility, structure, and strength. Its specific function is going to depend on what the cell type where this production is taking place. Actin can be found in most eukaryotic human cells, especially present in muscular cells. Thank you so much for watching.